So imagine you are sitting in the emergency room. You've got a sick infant under 6 months old, right?
Oh, which is incredibly stressful.
Yeah, exactly. It's terrifying. They have a severe infection, and the attending doctor prescribes a life-saving course of intravenous antibiotics. And, you're probably feeling this massive wave of relief that treatment is finally starting.
Right, because antibiotics in those situations are absolutely critical.
Exactly. And we all know antibiotics can disrupt the gut bacteria, right? You might even ask the doctor about starting a probiotic to help replenish those good bacteria once you take the baby home. But there's this one thing that almost no one asks about.
Not even the specialists, usually.
Right, not even the specialists, and that is the fungi.
It's true. The focus is overwhelmingly binary. I mean, we look at the bacteria because those are the known variables. We've been studying them for decades. But the fungi, which share that exact same physical space in the gut, they are practically invisible in standard clinical discussions.
Which is wild.
Yeah.
So today, we are doing a deep dive into some brand new research. This is from a June 2026 paper, and it finally connects these dots. It turns out, by wiping out the bacteria, we might be accidentally rolling out the red carpet for fungi.
And setting the stage for childhood asthma, which is a massive clinical revelation.
Yeah, we are going to map out exactly how an imbalance in the gut's fungal community, what's called the mycobiome, actively rewires the immune system and causes inflammation all the way up in the lungs.
And what's really compelling about this specific research is that it moves past just simple observation. We've known for a while that early life antibiotic use is a risk factor for atopic diseases like allergies and asthma.
Right.
But this study isolates the microbiome. It traces the exact cellular mechanisms that turn a localized gut disruption into a systemic respiratory disease.
Okay, so to give you a visual framework to hold on to as we get into all this biology, because we're getting deep into the weeds, I want you to think of the infant gut as this bustling, dense rainforest.
I love this analogy.
Thanks. So the bacteria, they are the towering trees holding the whole structural ecosystem together. And the fungi, they are the small ferns on the forest floor. They're living in the shadows, competing for, whatever tiny scraps of sunlight and water make it to the bottom.
They're kept in check by the big trees.
Exactly. But when a wildfire comes through, which in this case is a broad-spectrum antibiotic, it burns down all those towering trees. So suddenly, those little ferns have unrestricted access to all the sunlight, the nutrients, and the space they could ever possibly need.
And they just grow completely out of control.
Yeah, they take over.
And the central question of this study becomes, what does an unchecked overgrowth of those ferns do to the soil, the local wildlife, and just the entire ecosystem?
Right, the collateral damage.
Yeah.
So to understand the clinical reality of this, we first have to look at what actually happens in a human infant's gut when that antibiotic wildfire hits.
Which brings us to the antibio study.
Yeah, let's break that down. The researchers started with this observational clinical cohort. They tracked 47 infants, all under 6 months old, right in the emergency department of the Alberta Children's Hospital.
And they divided these infants into two distinct groups based on how long they were treated.
Right, so you had a short-term group of 22 infants who got antibiotics for 2 to 3 days, and then a long-term group of 25 infants, getting them for 4 to 14 days. And that long-term group included both the IV antibiotics in the hospital and the oral ones they kept taking at home.
So to measure the actual impact on the microbiome, you can't just look under a standard microscope. It's too complex. You have to measure their genetic footprints.
Yeah, how exactly do they do that?
Well, they used targeted genetic sequencing. For the bacteria, they used a technique called 16S rRNA sequencing.
Which is like a barcode scanner for bacteria, right?
Yeah. Exactly like a barcode scanner. It looks for a specific gene unique to prokaryotes to identify exactly which bacteria are present. But for the fungi, they had to take it a step further. They used something called ITS-qPCR.
Okay, unpack that a little for us. ITS-qPCR.
Yeah, so ITITE stands for internal transcribed spacer. It's just a specific region of fungal DNA. And qPCR is quantitative polymerase chain reaction. So instead of just identifying the fungal species like a barcode, qPCR actually weighs the total fungal DNA mass.
Oh, wow. So it gives you the sheer scale of the population, not just a roll call.
Exactly. It tells you exactly how much fungus is physically there.
Okay. So when they analyzed those genetic footprints, the results were highly counterintuitive, especially for the bacteria.
They really were. In the short-term antibiotic group, they saw a massive drop in bacterial concentration, which is, exactly what you would expect when you take an antibiotic.
But the long-term group was weird.
Right. In the long-term group, the total bacterial biomass didn't actually drop.
Wait, really? They took antibiotics for up to 2 weeks and their bacteria didn't drop?
Nope. And it implies a rapid adaptation. Basically, when you apply sustained, heavy antibiotic pressure for that long, the sensitive bacteria die off quickly. But antibiotic-resistant strains of bacteria multiply incredibly fast to fill that void.
So the total mass rebounds, but it's just full of resistant bacteria.
Exactly. The mass rebounds, but the quality and the diversity of that bacteria are severely compromised. But honestly, the truly alarming metric wasn't the bacteria at all.
Right, it was the ferns.
Yes.
Yeah.
In both the short-term and the long-term groups, completely regardless of how the bacteria responded, the total fungal biomass just skyrocketed.
The ferns took over the forest floor.
Mm-hmm.
And they didn't just multiply in sheer numbers, right?
No, they didn't. The researchers used a statistical tool, it's common in ecology, called the Che-1 index.
The Che-1 index.
Yeah, it measures species richness. Basically, it looks at how many different types of species are in an environment. And they found that while bacterial diversity plummeted, fungal diversity actually went up significantly.
And out of all this shifting ecology, this one specific genus of fungus emerged as the absolute dominant force, right? Malassezia.
Specifically, they isolated 3 species that just took off: Malassezia restricta, Malassezia globosa, and Malassezia sympodialis.
Okay, wait a second. I got to stop you there because Malassezia, isn't that a skin fungus?
It is, yeah.
Like, it's universally known for causing dandruff. So what on earth is a dandruff fungus doing, surviving, let alone dominating, inside a baby's intestines?
It's a great question, and it comes down to the specific dietary environment of an infant. Malassezia is really unique in that it is lipid-dependent.
Meaning it eats fat.
Exactly. It completely lacks the ability to synthesize its own fatty acids, so it literally has to consume external fats just to survive.
And an infant's diet
is exclusively breast milk or formula. It is incredibly rich in lipids. The infant gut is essentially this warm, highly concentrated fatty buffet.
Oh, that makes so much sense.
Right. Under normal conditions, the diverse bacterial community consumes all those resources, rapidly and actively crowds the fungus out. But the moment the antibiotics wipe out that bacterial competition,
Malassezia gets exclusive access to the buffet.
Exactly. In fact, the data from the antibiotic study showed that the total number of antibiotics prescribed was the single strongest predictor of this aggressive fungal expansion.
So it's totally dose dependent. That is fascinating biologically, but it also fundamentally changes the structural architecture of the gut, right?
Absolutely. Think about the gut as a city network. Before the antibiotics, you have these highly beneficial bacteria like bifidobacterium and bacteroids.
The mayors of the city.
Right, the mayors. They are deeply connected to everything else in the network. They keep order, they maintain the structural integrity of the gut.
But post-antibiotics, those mayors are entirely eliminated.
Gone. And they are replaced by opportunistic pathogens. When the researchers ran a network analysis of the gut community after the antibiotics, the nodes representing Malassezia grew massive.
They became the new network hubs.
Yes. This wasn't just a fungus surviving in the background quietly. It was executing a hostile structural takeover of the entire microbiome network.
Okay, so let's take a quick second to recap where we are, because that was a lot of biology.
Good idea.
Basically, antibiotics act like a wildfire. They reduce bacterial diversity, which then allows lipid-loving fungi like Malassezia to aggressively expand in the fat-rich infant gut and literally take over the ecosystem.
That's the perfect summary.
And so, for you listening, here's a question to chew on. Have you, or maybe a child you know, ever had to take early life antibiotics? And if so, did you ever consider how it affected the fungi in the gut, rather than just the bacteria?
Most people never think about the fungi.
Never. Okay, so we have this clear chain of events in human infants, but observing this in a hospital brings up a classic scientific hurdle, doesn't it?
It does. It brings up the issue of causation versus correlation.
Right. We can see the antibiotics cause the fungal explosion, and we know kids who take early antibiotics have higher rates of asthma. But how do we know the fungus is actually causing the disease?
Exactly. What if it's just an innocent bystander enjoying the fatty buffet while the original infection or something else is what's actually causing the asthma?
Right. You can't prove causation in an observational human study. There's just way too many variables. Diet, genetics, the original infection.
Way too messy. So to prove that Malassezia actively causes harm, the researchers had to move to a highly controlled animal model. They needed an absolute blank slate.
Enter the germ-free mice.
Yes. also known as gnotobiotic mice.
Gnotobiotic. These are animals raised in completely sterile isolators, right? Like bubble boys. They have zero bacteria, zero fungi, zero viruses on or inside them.
Correct. They are a true blank slate. So the researchers took these germ-free mice, specifically a strain called C57BL6J, and colonized them with a very specific known cocktail of exactly 12 bacteria.
Just 12.
Just 12. It's called the oligo-MN12 cocktail. It's a standard protocol used to simulate a healthy baseline gut microbiome, so they have a control group.
Okay, so they established the baseline. Then what?
Then they introduced the critical variable. They created a second group of mice with that exact same 12 bacteria baseline, but they added our antagonist, the fungus Malassezia restricta.
And the way they delivered it is wild. They didn't just inject the mice.
No, they wanted it to be as natural as possible. They smeared fresh fungal cultures on the mother mouse's abdomen.
So the pups would naturally ingest the fungus while nursing?
Exactly. It perfectly mimics how human infants acquire their early microbes from their mother's skin and milk.
That is incredibly elegant experimental design. Because they controlled exactly what microbes were in the gut, any changes in the health of the mice had to be directly caused by the addition of that one specific fungus.
Yes, it isolates the single variable perfectly.
So what happened?
The changes were profound. Even though the fungus was only present in relatively small amounts compared to the bacteria, it totally shifted the environment. Certain bacteria like E. faecalis increased, while beneficial ones like Limosolactobacillus reuteri decreased.
Wow, just from adding one fungus.
But the most striking evidence came when they looked at the chemical footprint left behind in the gut. They used a technique called untargeted metabolomics.
Okay, metabolomics. Instead of looking at the microbes themselves, this scans for all the small molecules and metabolites left behind by their cellular processes, right?
Yeah.
Like looking at the trash left behind to see what they were eating.
Exactly. And when they scanned the gut for chemical changes, they found a massive 6.56-fold drop in a molecule called oleate.
Oleate. Wait, oleate is an anti-inflammatory fatty acid, right?
It is, and it's naturally highly abundant in breast milk.
Oh, so if Malassezia is a lipid-dependent fungus that needs to eat fat to survive, it is literally consuming the infant's anti-inflammatory defenses.
That is the leading hypothesis, yes. By consuming the oleate, the fungus removes a critical mechanism that keeps the gut's immune system calm.
It's eating the peacekeepers.
Exactly. And the chemical changes didn't stop there. The researchers also saw drastic drops in the stress hormones cortisol and corticosterone, which also play significant roles in regulating immune responses.
This single fungus was entirely rewiring the biochemical landscape of the gut.
Completely. And with the anti-inflammatory fats depleted and the regulatory hormones dropping, the immune system is obviously going to react.
So where does the immune system mount this defense?
The researchers focused on two specific anatomical locations to watch this unfold. First, the colonic lamina propria, or CLP.
That's the tissue lining the gut itself.
Yes. And second, the mesenteric lymph nodes, or MLN, which basically function as the local immune headquarters.
Okay, so when the immune system detects this potential fungal threat in the gut lining, it deploys these highly specialized units.
Right. In the mice exposed to Malassezia, researchers observed massive spikes in two specific types of T helper cells, Th2 and Th17 cells.
Okay, I actually have a mnemonic for this to help keep these straight because immunology can get confusing.
Oh, let's hear it.
The OCT2 brings the ACHU, Th17 brings the scene.
I love that.
Right. Because Th2 cells are heavily involved in driving allergic responses. The ACHU and Th17 cells are specialized in fighting fungal infections by triggering intense, localized inflammation. They cause a scene.
That is a perfect way to remember it. Both of these T cells spiked dramatically in the gut lining and the lymph nodes. And alongside those T cells, there was a huge afflux of eosinophils.
Eosinophils. Those are the white blood cells that specifically target multicellular parasites and fungi. But, and this is the key, they are also the primary cells infamous for causing the severe inflammation seen in allergic asthma.
Exactly. We also saw a spike in macrophages and migratory dendritic cells.
Okay, the migratory dendritic cells are so crucial here. You can think of them as the scouts of the immune system.
Yes, the communicators.
Imagine those dendritic cells as sentries patrolling the border of a kingdom. They spot the invading fungus in the gut, grab a physical piece of it, an antigen, and literally travel all the way back to the castle.
Which is the mesenteric lymph node.
Right. They present this antigen, effectively waving a red flag and signaling that the kingdom is under attack. In response, the castle begins producing a massive, aggressive standing army of those Th2, Th17, and eosinophil soldiers.
And having a massive standing army with nothing to fight is exactly the problem.
Right, they're just waiting around.
The immune system is now hyperactivated. It is highly localized in the gut, but it is primed, agitated, and poised for a massive inflammatory response.
Which brings up a really compelling thought. If you have this single fungus completely rewiring the local immune system to be on high alert, how might this agitated standing army misbehave if it encounters something completely harmless in the everyday environment?
Like household dust.
Exactly. Okay, so time for another quick 5-minute recap.
Let's do it.
By putting Malassezia into germ-free mice, we see it consumes anti-inflammatory fats, like oleate, and it triggers an aggressive immune response in the gut and lymph nodes. Specifically, it spikes allergy-linked Th2 cells and eosinophils. So, listener, here is your question to ponder. Knowing that this single fungus can completely rewire the local immune system, how do you think this massive standing army of immune cells might misbehave when it hits a normal allergen?
And that bridges us directly into the concept of the gut-lung axis.
The gut-lung axis. Because we've built this angry, primed immune system in the gut, but asthma happens in the lungs. How does a gut fungus cause a lung disease?
Right. The immune system is not strictly compartmentalized by organ. Cells, chemical signals, and inflammatory markers, they travel systemically. So the researchers had to see if this primed gut would translate to respiratory disease.
So to test that bridge, they used something called a house dust mite challenge.
The HDM challenge, yes. They introduced dust mite allergens directly into the airways of the mice, basically simulating what happens when a baby breathes in common dust in a living room.
And the reaction was severe.
It was. The mice that had the Malassezia fungus in their gut developed violent lung inflammation. When researchers did a bronchoalveolar lavage, which is washing out the lungs to analyze the fluid, they found it was absolutely packed with eosinophils.
So the fungal imbalance in the gut had successfully primed the lung tissue to aggressively overreact to plain old dust.
Exactly.
But wait, I'm going to push back on this for a second. If the eosinophils are the primary cells flooding the lungs and driving this allergic reaction, what happens if we just take them out of the equation entirely? Does the asthma problem completely go away?
That is the exact critical question the researchers asked, and to answer it, they used a highly specialized genetically engineered mouse mutant. It's called the delta debilitate1 strain.
Okay, that's a mouthful. What does that mutation do?
It's a specific strain bred to have absolutely zero eosinophils in its body.
Oh, wow. So they literally deleted the eosinophils.
Yep. Right. And they ran the entire experiment again. They took these germ-free eosinophil-deficient mice, added the baseline bacteria, added the Malassezia fungus, and exposed their airways to the dust mites.
And the results were super nuanced, right?
Very. Removing the eosinophils did successfully stop the macrophage response in the gut.
Okay, so a partial win.
But, and this is the huge but, it did absolutely nothing to stop the Th2 cells, the Ta17 cells, or the severe lung inflammation.
Wait, so they had zero eosinophils and they still got the asthma-like inflammation?
Yes. It is a critical finding because it proves that while eosinophils are a major component of the allergic response, they are not the sole drivers.
It's the T cells.
Exactly. The adaptive immune system, those highly specialized Th2 and Th17 cells, is fundamentally reprogrammed. It drives the asthma response completely independently of the eosinophils.
So it tells us we are looking at a deeply entrenched, multi-pronged systemic immune malfunction. It's not just a simple overproduction of one white blood cell type.
Right. The fungus essentially creates a resilient, multi-layered allergy machine.
Which is terrifying. But, human biology is rarely that isolated. Infants aren't just exposed to dust. They're constantly bombarded by viruses.
True.
And the researchers knew that, so they introduced a final layer of complexity. They used a different mouse strain, the BALBC strain, which naturally produces a more mixed, complex inflammatory profile, and they introduced respiratory syncytial virus.
RSV, which is incredibly common. I mean, almost all children get it by age 2.
Right, and early severe RSV infection is a well-documented risk factor for developing asthma later in childhood anyway.
Exactly. So they infected these infant mice with RSV at 7 days old to see how the virus and the fungus interacted.
Now, surprisingly, having the Malassezia fungus in the gut didn't actually make the initial RSV viral infection any worse.
It didn't. The infant mice fought off the acute viral infection normally.
But the lasting damage is what matters.
Yes, and this leads us to the two-hit hypothesis.
The two-hit hypothesis. Break that down.
Well, When those same mice grew up and their airways were later exposed to the house dust mites, the combination of having survived early RSV plus having the active gut fungus resulted in a devastatingly severe mixed inflammatory response in the lungs.
So not just eosinophils this time.
No, we saw massive influxes of neutrophils, lymphocytes, and monocytes. It was a completely exacerbated reaction.
So it's a compound effect. The virus acts as the first hit, structurally damaging and sensitizing the lung tissue during a vulnerable developmental window.
Right.
And the gut fungus acts as the second hit, relentlessly building and maintaining that hyperactive immune army we talked about.
Precisely. When you combine them, they virtually guarantee severe allergic inflammation the moment the lungs encounter a common allergen.
It creates a perfect storm. The viral infection lowers the threshold for inflammation in the airway, and the fungal dysbiosis provides the systemic immune ammunition to just exploit it.
It's a profoundly integrated ecosystem, and honestly, it completely reframes how you view the interconnectedness of your own biology.
Totally. I mean, we are conditioned to think of our bodies as a collection of separate organs, right? The gut processes the food, the lungs process the air, and they kind of just mind their own business.
But this shows that an ecological shift in your intestines can dictate exactly how your lungs respond to the air in your living room.
It's mind-blowing.
Okay,
Let's do our final 5-minute recap here.
sounds good.
A fungal imbalance in the gut primes the immune system so intensely that when the lungs encounter normal allergens like dust or common viruses like RSV, the body launches a massive, damaging inflammatory attack.
It's the two-hit hypothesis in action.
Right. So for you listening, we usually think of our body's systems as completely separate. How does this gut-lung axis change the way you view your own body's interconnectedness?
It definitely makes you look at the whole picture differently, which brings us to a vital synthesis of everything this research represents.
Yeah, let's bring it all together.
Yeah.
We need to be really clear here. The infants in the emergency room during that initial antibiotic study, they had severe, potentially life-threatening infections.
Yes, they absolutely needed those intravenous antibiotics. We cannot overstate that antibiotics are vital, life-saving tools. We are not saying people shouldn't take them.
Right. Definitely not taking a stance against necessary medical treatments. We're just reporting the biological mechanics found in the source text.
Exactly. The big picture takeaway here isn't a simplistic antibiotics are bad.
No, the takeaway is that we have to acknowledge and manage the profound ecological collateral damage they cause. We are saving the patient by burning down the bacterial trees in our microbiome's rainforest.
But we are blinding ourselves to the Malassezia ferns that thrive in the aftermath.
Beautifully put. And we now have the mechanism showing that this specific fungal overgrowth consumes anti-inflammatory defenses, it triggers a localized immune-standing army, and it fundamentally rewires a child's developing immune system toward lifelong atopy and asthma.
It challenges the entire modern medical paradigm. Treating an infection isn't just about neutralizing the immediate pathogen anymore. It has to be about actively managing the long-term ecology of the host.
Which leaves us with an incredibly compelling, almost provocative thought regarding the future of medicine.
What's that?
Well, if early life asthma is so heavily driven by this fungal overgrowth following necessary antibiotic use, the way we treat childhood infections might look completely different in 10 years.
Oh, absolutely.
Right. Like, could you see a world where a pediatrician prescribes a life-saving antibiotic, but alongside it, they administer targeted antifungals to keep Malassezia from taking over?
I could definitely see that. Or perhaps instead of just typical bacterial probiotics, they prescribe specific profungals to restore a healthy, competitive microbiome balance.
That would be wild. The future of medicine will likely demand that we treat the entire microbiome, fungi included.
Once you recognize the complexity of the whole ecosystem, you just can't go back to ignoring half of it.
You really can't. So next time you hear about a frustrating, stubborn case of childhood asthma, I bet you won't just picture the lungs. I bet you'll think about that microscopic rainforest, the missing trees, and the ferns quietly taking over the forest floor. Keep digging, keep questioning, and we'll catch you on the next deep dive.